Okay, this is cell communication part 2 of our lecture, and we're going to ask you to type in the chat an answer to this question, but don't hit enter right off the bat. We kind of want to give people a chance to type. We all sort of think and type at different speeds, so don't feel like a rush to put enter. I kind of like you to hold off on hitting enter because I want to see what you think, not just, well, that guy said it, so I'm going to say it too. So go ahead and put this in the chat, what you think, some ways that cells process signals. So yesterday we talked about the 3 ways. They do direct contact, we do local and long distance. This is like, well, what happens once they get the signal? So type something in the chat. This also lets me know that you're actually present here and that you're, engaged in class. So I like to see everybody participate in these. All right, go ahead and hit enter if you got something written in there. Okay, see the word receptor coming up a lot, and that is true. Okay, so we do have a lot of types of receptors. Okay, so I'm glad to see that. It tells me that we have maybe some familiarity with this, maybe some experience with this. All right. All right, if you haven't gotten something in, go ahead and do it now, and we'll go ahead and begin. Too many windows. So if you said something about receptors, that's a lot of what we're going to talk about. I see the word transduction, someone said out there. Receptors process the signals. They use receptors. A lot of people definitely have this idea of a receptor down, and that is the sort of the first part we're going to talk about. So this idea of cell signals, we're going to break it down into 3 stages, okay? And we're just going to kind of skim the surface. These stages can actually be way more complex than sort of our big view that we're going to present. Okay, so first word we're talking about, that green word there that's important to you is reception. We're going to turn receptor into a verb. So we have what's called a ligand. So a ligand is probably a new word for you. And a ligand is just some sort of signal chemical. In this case, it's these orange molecules that are coming in here. Those are ligands. So ligand's probably a new vocabulary word for everybody. Know what a ligand is. Sometimes ligands are hormones. So there's different kinds, and these ligands are going to chemically bond to a receptor. Okay, so we all understand that. We've all probably seen examples of that. The next step, which I did see pop up in the opener question, is transduction. So transduction is where the signal is converted into some process, and that's usually, in this one right here, it's only, a couple steps. But we see these 3 changes. So it's 1, 2, 3. In actuality, these can be up to, around 100 steps. Cell signaling, very complex. When it breaks down, a lot of diseases are related to broken cell signaling, but we flip the switch and then something happens. That's what transduction is, okay? If you think about it, when light hits your eyeball, what you see doesn't really happen in your eye, okay? Light hitting your eye in your retina in the back, that's reception. Then that signal is converted to a nervous system process, a nervous system signal. That's transduction. So it's not like light goes onto your brain like a camera sensor or something like that. What you're actually, the images that we perceive happen after reception. Okay, and the final step is a response. Okay, so you might see something and respond or not respond to it. So that response might be like, well, that's pretty interesting, I'm going to pay attention to it. That response might be, that baseball's going to hit me in the head, I'm going to duck and close my eyes. So I'm trying to give an analogy to this on a bigger biological process. So on the cellular level, some process is altered. That could be turning off something the cell does. That could be turning on something the cell does. Usually making a new protein or opening channels to either absorb or release something. Those are some very common responses. Make something new, open up and let something in, open up and let something out. Those are common responses. There can be lots of other ones, like for muscles, it could be move. Okay, so we gave you those 3 steps: reception, transduction, response. Now we're going to talk about each one in greater detail. Okay, so for the first stage, reception, what's going to happen? Reception is the detection and receiving of a ligand. Okay, remember, a ligand is some sort of substance that hits the receptor. There's lots of different kinds of those. We'll talk about that a little bit later. So, what are receptors? Okay, receptors are big molecules, usually proteins, and they will bind to some sort of signal molecule. And again, signal molecule, ligand, same thing. I know I'm repeating it a lot, but that's what teachers do when we introduce new words. So all receptors have a spot, almost like an enzyme has an active site. It's not exactly the same, but it's similar. And that spot on the receptor, when the ligand makes contact, it's going to somehow change the state of that protein. So the binding is very, very specific, okay? We don't see, the ligands for one cellular signaling pathway turn on, the wrong one usually. Now, different drugs can act as ligand or ligand inhibitors. Different toxins can do the same thing. We can block or overload cellular pathways, either, with the actual original ligand, but there's also ways we can fool these and make them do what we want to do with drugs, or we can also do bad things to them with toxins. But it's very specific. So it's almost like an enzyme. Enzymes have very specific substrates they work on. Okay, so when the ligand bonds, here we have these orange ligands headed for this receptor protein on the cell membrane, some sort of change in the shape of that receptor is going to start the next step, and that's transduction. So this is basically turning on the switch, and then transduction is sort of the electricity moving through the wire. Now, this is kind of a big deal right here, okay? So this section right here, I want you to kind of pay attention to. In my picture, the receptor is on the membrane. But there are receptors that are also intracellular. They can be on the surface of an organelle. They can be floating in the cytosol, but there are different receptors in different locations. Okay? The authors of AP Test seem to have a lot of reason to pay attention to this. So you're going to probably see AP Test questions. They're going to ask you, they're going to give you some ligand you've never ever heard of before. They're going to give you some receptor you've never heard of before. And they're going to tell you something about either the ligand or the receptor, and see if you can. They're basically going to ask you a question in such a way that if you can tell if it's on the membrane or if it's intracellular. So I'm going to give you the ways to tell the difference. So, all right, this is kind of a big deal, so make sure you're understanding this. Plasma membrane receptors are the most common. Now, that's not how you tell them apart, but they are more common, okay? And that's what most of your biology education has shown you so far. And that's why my first example was something you're probably familiar with, a receptor embedded in a phospholipid bilayer. So they are the most common. So they are going to tell you that a ligand, they'll probably tell you, they're not going to say, well, here's the ligand, figure out if it's water soluble or something like that. They're going to tell you that. So when you see that a ligand is polar, water soluble, or it's big in size, it's going to be plasma membrane receptor. And from what we've learned about plasma membranes, this shouldn't be shocking. Now, you've learned a lot this year, and you may have I remember something about that, but I need a review. That's why I'm really taking my time right now, because what you're going to see in this course is the stuff you learned earlier is going to be foundational to these next steps. And they're going to ask you questions in such a way that they can figure out if you know stuff from Unit 1, 2, and 3 all in one question. If you've ever taken an AP test before, you probably have seen that, okay? Now, if you remember plasma membrane, phospholipid bilayer, big stuff doesn't go through very well. So this should make sense. Water-soluble stuff can go through, but very, very slowly. So a lot of times we don't want signaling to happen slowly. Polar things don't go through very well because they're going to get caught on either the hydrophobic or hydrophilic end with that polarity. So this is a review of the properties of the phospholipid bilayer and what kind of things can and how things interact with it. Now remember, they're not going to say to you, here is this ligand you've never heard of. Is it polar? They're going to tell you the polar, water-soluble, and large stuff, okay? So here are some examples of these that we'll get into later. G protein coupled receptors. That's something we're going to get more on down the road. Ligand gated ion channels. When some ligand bonds with one of these receptors, it will send a signal to open, like we already talked about, to open or close a transport protein, basically. All right. Intracellular receptors, the ligands have to be able to move through the plasma membrane, the phospholipid bilayer, to get in the cell. So we got to think back to what are the properties of substances that can get through the cell. Well, it's pretty easy because it's the opposite of the others. Okay? Where are these receptors? They're either, like I said, in the cytoplasm. A lot of times they're in the nucleus because your cells might get a signal, start making this stuff. Well, if I make this stuff, I need to get the DNA code for that protein of what I'm supposed to make. So a signal will land on the nucleus that says, send out the plans for such and such protein. And that's how we turn on the production of a certain substance. You know, if you go donate blood, your body's going to need to replace those blood cells. So you're going to get a signal, it's picked up in your kidneys, that says, I'm low on blood, make more. So certain cells in your bone marrow will start making more blood. So that's just an example. But where's the plans? Where's the plan for making red blood cells? They're on DNA. So a lot of times these signals head for the nucleus. It can be other organelles. These are the super common ones. Okay. So intracellular receptors are going to be receptors that pick up and bind to ligands that can get through the membrane. They are going to be hydrophobic, and they could be things like steroids and thyroid hormones, and also gases. Some gases can move through the membrane. It says LE hydrophobic there. I think I want that to be non-polar. So add non-polar there. Let me see if I can do that for you. All right, that was a lot. Okay, how are we doing out there? Give me I'm okay, or is my pacing okay? Give me a speed up, slow down, or it's okay in the chat, just so I know that you're out there.
Your pacing's fine.
Thank you. Everybody, I'm looking for a chat, just so I know you're still at your desk. All right, so a lot of times we sit in class and we see things in lectures and we don't want to go back and look at them. Try to review this sometime, not right now, in the next 24 hours. Come back and look at this, whether it's my video, whether it's the slides I've posted, or whatever. Just come back and read through this, okay? There's a lot of stuff here. Okay, let's look at this a little bit closer, okay? So here's the cell membrane, and this purple part here is supposed to be the nucleus, okay? So this particular signaling molecule made its way into the cell, into the cytoplasm. It's going to bond with some sort of receptor that's present in the cytoplasm. That's going to interact with the nucleus and tell a specific gene to be expressed. Make this protein. Okay, and that could be, this is very complex stuff, okay? So, what we've got down here at the bottom is important to note. I've said it already, but I'm going to say it again. The AP exam people, they don't expect you to classify molecules. They're going to tell you, but they might say it's a steroid. So if they tell you it's a steroid, steroids can go through membranes, okay? You can get topical steroid creams to put on your skin, and that's one reason why you can get topical steroids, is because they can go through membranes. So if you want to go into pharmacology, this kind of stuff can be important, because like if you're out there saying like, well, we got to get a drug that does this, well, do we have to give it as a something that they put in their mouth orally, like a pill? Can we give it as an injection, or can we make it into a cream? And so there's, a lot of implications for this. That's just one of them. All right, let's look at transduction, okay? So transduction is converting the signal to some sort of response. We're going to convert a signal to a response. So you're going to see a set of changes or a chain reaction. If you want to think of dominoes falling down or one of those like Rube Goldberg machines, that's kind of a good analogy to have in your mind, where the receptor protein is going to set something else off. Now, those purple words I have there, signal transduction pathway, try to use that exact phrase in questions you're going to answer about this sort of stuff. The signal transduction pathway is going to be limited by the absence of this. The signal transduction pathway will break down because of a mutation in that protein. The signal transduction pathway will be activated by, learn to use the word signal transduction pathway. That's something that graders are going to be looking for on the exam. Signal transduction pathway. Okay? Kind of tattoo that on the biology part of your brain right now. Signal transduction pathway. And I want you to know what it is. A signal transduction pathway is, like I said, a series of changes. A lot of times in biology, we think of like, this makes that happen. But usually from here to there, there's lots of little steps in the signal transduction pathway. And this is why biology is so complex, because there's usually intermediaries where things if one little change along this signal transduction pathway happens, it can break down your cellular responses and lead to disease, illness, bad things. All right.
One moment. Hello.
Siri wants me. Okay. So what's actually happening? Here's some new terms I want you to learn. The signal transduction pathway regulates protein activity. Okay, well, what does that mean? That means when a protein's shape is changed or state is changed, it happens through these two enzymes here. So I want you to know these. The protein is going to be phosphorylated. Where's the phosphate coming from? Most likely ATP. By an enzyme called protein kinase. Okay? So I want you to write in your notes, this one is the on enzyme, and this one down here is the off enzyme. Okay? So when we're going to turn on proteins through a pathway, it's going to be this enzyme, protein kinase, by phosphorylation. We're going to be adding phosphates to it that are going to change its state, and then it's going to send some sort of, it's going to be a link in this signal transduction pathway, okay? So if you think about walking down a pathway, each step of it turning on is going to be a protein kinase, okay? And listen, there could be lots of things that, like, all these things, like say you come up to an intersection and you want to go down the road. Many things have to be true before your car will roll down the road, okay? First of all, the red light, okay? That could be step one of the pathway, okay? If it's red, don't go, okay? So light goes red, or the light turns green. So you're kind of like, well, that's going to be the ligand that says go. But a whole bunch of things have to happen, exact right order, for you to proceed. First of all, if you're a good driver, even though the light's green, you're going to look around, make sure it's safe. So that might be step one of phosphorylation. The next step, if that step proceeds, would be my foot is going to push my foot's going to come off the brake. That's a step. Okay, so that would be another phosphorylation. The next step might be my foot is going to press the gas. That's another step of phosphorylation. And so on and so on and so on. And so if any one of those gets disrupted, the pathway should stop. Okay? And so those could be biological things that need to be true for this pathway to be useful or helpful or appropriate for homeostasis. Okay? Once all this stuff happens, you're driving down the road, you see another red light. Dephosphorylation starts doing this backwards usually, and all these things are going to happen with an enzyme called protein phosphatase. Okay, you got that ACE there. And this is going to dephosphorylate, take the phosphate out, okay, and shut this off. You don't have to know any specific ones, but there's some like really popular examples we're going to look at, okay? So what we're going to want you to be able to do as students is they're going to show you one you've never heard of before. They're going to probably disrupt it somehow and ask you to predict what's going on here. So you should be using the words signal transduction pathway is going to be turned on by phosphorylation. Okay? Signal transduction pathway is going to be turned off by protein phosphatase. Signal transduction pathway is going to be disrupted by the absence of or the presence of this toxin, mutation, disease, whatever. So those are the kind of targets I want you to be able to hit after getting through this unit. This is some heavy stuff. This is the first time you've probably heard of this unless you're, I don't know, some biological prodigy that's reading the textbook all night long. Most of us aren't that way. So this is something you're probably going to have to study a little bit more than normal, okay? Remember, it's supposed to be a college class. You don't just hear it once in class and say, I'll get it on the test. You're going to have to review a little bit of this on your own, watch some videos, study it, find your own way of remembering it. Okay. So any change in the shape of one of these proteins, this is a big theme in all of biology. Changing the shape of something changes its function. So all these phosphorylations or dephosphorylations, you add the phosphate to the protein and it somehow changes. You take the phosphate away and it somehow changes back. So shapes and functions are very important. If you were going to put two Lego blocks together and someone crushed it, it won't go together anymore. Okay? Changing its shape is very important to its function. Okay. Usually, during transduction, the signal is amplified. Okay, so what does that mean? That means that one ligand molecule is usually going to activate multiple subsequent steps of the pathway. So this is like, have you ever seen people set up the dominoes and they set up 1 and then 2 and then 3 and then 4, where like one domino kind of spreads out and knocks a whole bunch down. So we're amplifying the pathway by the first thing is going to turn on a whole bunch of other things, okay? The first thing is going to turn on a whole bunch of other things. Now, don't think of the amplification as, well, there's more ligand, okay? It only takes one switch to turn on, and then a whole bunch of the second step will happen. And then maybe the third step turns on even more. So this is where you want, in biology, one tiny little signal to have a huge effect, okay? It's not usually... It can be. It's not usually a more ligand, more. It only might take a very tiny amount of ligand to get this big change in your body. An example of that is when someone's having an allergic reaction, one little shot of epinephrine can change the whole state of the whole body. very, very quickly. That signal, that epinephrine, to open up the airway, open up blood vessels, that signal is amplified in the body very, 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 very quickly. If that signal had to get to every little cell, that would not be a very fast-acting drug. Okay, a lot of these drugs can act very fast because of signal amplification. All right, finally, okay, our third stage is going to be response. So the response is some sort of alteration, something that cell wasn't doing before, it's going to start doing or stop doing. So that's usually make something, stop making something, open up, move. It could be a lot of different things, but those are the common ones, okay? So here's where I wrote down the examples, okay? Some protein can change the permeability of the membrane. Okay, cells, we need to release this signal, okay? That happens during blood clotting. Okay, cells, we need to take in water. This can happen to plants during different types of times that they're exposed to water. Okay, some desert plants don't get water very often, and when they do, they want to take up as much as they can. Okay, cells make this enzyme. Okay, we need to change our metabolism because we've loaded up on sugar, we've loaded up on protein, we've loaded up on fats. So if you sit down and eat a really fatty meal, let's say you have like French fries and a big fatty steak or something like that, your digestive system is going to need the materials to break that down. So telling cells to make those would be a response. This next one, gene regulation, is a really big deal, okay? Imagine a fetus is developing. There are certain proteins that are only turned on for very, very short times in fetal development. There's proteins that make enzymes that change the structure of your brain if you're male or female. Those aren't on all the time. Those are only on or during very, very limited times of fetal development. But it can be everyday stuff too. It can be, hey, if you over shampoo your hair, your oil glands on your head make more oil because you're constantly stripping the oil off your scalp. So you're going to have a protein that turns on the gene to have more oil produced in your oil glands. So it can be little stuff too. It doesn't always have to be like these profound biological changes. It can be as simple as, does my hair get oily or not? Okay. So responses are all over the place. As biologists and as, healthcare providers, understanding these pathways is how we unlock the understanding of disease, okay? So are these pathways broken? Are these pathways overproducing? Or are these pathways missing something? And so this can be a really, really big part of medicine, okay? And, the average person who maybe diagnoses a disease doesn't have to know every single one of these. They just have to know the symptoms and the treatment. But the people who are actually, back in the lab looking through microscopes, this is the kind of stuff they're doing experiments to understand better. All right, let's do a quick review. I want you to type these, okay, just like we did yesterday. Give me those 3 stages. That was a huge theme. So just give me a 1, 2, 3 and hit enter in a minute. Not right now. I'll tell you when to hit enter. Everybody should be opening up the keyboard, okay? So I want the 3 stages of cell signaling. You got to tell me what those are. That was sort of the big opening theme, okay? A lot of you mentioned this in your warm-up question. All right, I'm just looking for 3 words. If you've got it, go ahead and hit enter. Okay. Yeah. For the listen, I am really looking for more than like 5 people to be answering. Yeah. Thank you. The rest of you. I don't really care how fast you are. Just let me know you're out there. Okay. If you don't have your camera on, you got to do keyboards. Okay. If you're not answering these, I'm assuming you're out. Getting a Whopper at Burger King or something. All right. So you guys are right. Reception, transduction, response. Okay. Remember, one of the big phrases I want you to sort of attach to this is signal transduction pathway. Signal transduction pathway. All right, next review question. Let's have 100% participation. There should be 23 answers coming in. All right. What's the actual signal that Mr. Sater mentioned today? What's the actual signal? I don't care if you're right or wrong. I just want to see your participation. It's not like this is going in the grade book, okay? You can look through your notes if you want to. All right, give me a answer. Go ahead and hit return. All right, I'm looking for ligand. Okay? Very good, guys. Thank you. All right, next review question. This answer's going to take a few words. All right, go ahead and shoot me those responses. Go ahead and give me some answers for that one. Somebody be brave and hit enter. Okay, so I'm looking for the word transduction. Okay, looking for the word transduction here. Yeah, okay. Yeah, this one was a little harder to kind of like, maybe I probably didn't give you enough time for that one. Yeah, so yeah, there we go. Signal transduction pathway. I'm looking for that too. When in doubt, signal transduction pathway. Okay, a couple more things on signal transduction pathways. So we kind of mentioned this, but let's look at it in just a bit more detail. Okay? Signal transduction pathways basically tell a cell how to respond to the environment. Everybody knows that, we can take insects or worms or goldfish and we can put things in the environment that are going to change the way they respond. Okay? If you have goldfish in a bowl, when the oxygen goes low, they're going to change the way they respond to their environment. They're going to start gulping air. So that's an example on an organism level. We can also see this on a cellular level. Cells will change the way they do things based on what signals they're getting. Okay? So it can either change the genes they express. An example of that is if you take yeast. This is a really common biology experiment. Biology teachers will hand out little samples of yeast, okay? Then they'll hand out a bunch of different sugars. They're going to give table sugar, they're going to give fructose, they're going to give lactose, they're going to give maltose, and each yeast will get a different type of sugar. They can all digest those sugars, but they have to make different enzymes to do each one. So when yeast digest things, they make CO2. Students will measure how much CO2 is made, and they'll say, well, yeast is really good at this sugar, but not so good at this sugar, okay? And so what we see here, how does the yeast know how to deal with the different sugars? Well, a signal transduction pathway says, all right, maltose is present. Make the enzyme that breaks down maltose. If the yeast cells were making every type of enzyme for every sugar all the time, that would be a huge waste. Once the sugar isn't there anymore, or if the sugar changes from maltose to lactose, the yeast changes its gene expression. Okay? This is really cool. I'm really hopeful to actually do my favorite lab of the year, where we put jellyfish DNA into bacteria and we give the bacteria a certain sugar that says express this gene, and then the bacteria glow in the dark. Very, very cool stuff. Okay? These signal transduction pathways can do a lot of different things. Okay? They can tell cells to die, and when that doesn't work, we call that cancer. So uncontrolled cell growth. Okay? So very, very cool that we can have cells respond to the environment and make different things. This is if you want to go get a tan, this is what tanning is. Okay? If you're, Caucasian, very pale skin, you get out in the sun, your body says, man, that's a lot of sun. Let's respond to the environment by making more pigments. That's what tanning is. The old fashioned, not the spray. All right. So, anything that changes in these pathways, mutations, okay, toxins and drugs, anything that changes it is going to basically interfere with the cell's response. Okay, so this is your little assignment for today. I'm going to go ahead and stop my recording and talk about what I want you to do.